Hello, everyone. Welcome to the TEAS Science Section workshop presented to you by the ARC. Today, we will be going over some concepts that you need to review for the science section. So the section is 30% of the total exam. This is just a question breakdown and timing. So 50 questions are given, and you're given 60 minutes to complete them. That's a little over 1 minute per question, which doesn't seem like a lot of time, so make sure you are pacing yourself properly. Some questions you will get through a lot faster than others, so just keep that in mind. There are 18 questions on anatomy and physiology, 9 questions about biology, 8 questions from chemistry, and 9 questions regarding scientific reasoning. The major topics covered are biology, anatomy and physiology, chemistry, and there's a bit of ecology as well. Now, just some general science know-how. So the metric system, we need to know how to convert between a base unit and units with prefixes, or between units with prefixes. So over here on the right-hand side, we have this blank row. This represents our base unit. An example of a base unit is meters, all right? If we wanted to convert meters to decimeters right above it, we would divide by 10 or move the decimal one place to the left. This is the same thing as multiplying by 10 to the negative first power. If we wanted to convert the base unit, in this case meters, to decameters, we can multiply by 10 or multiply by 10 to the first power, or move the decimal once to the right. This order of prefixes here is very important. If you need an acronym to remember this, King Henry died while drinking chocolate milk with 3 monks and 3 nuns is a very helpful one because the order of this definitely matters because each prefix can be matched up with an exponent that you need to multiply by in order to get your desired unit. Ready. Next is the difference between accuracy and precision. So accuracy is how close the values are to the true value. On the left here, we can see that these black dots are not very close to each other, but they are pretty close to the center. Whereas over here on the right, precision is how close the values are to each other. These black dots are very close to each other, but not close to the center. So remembering the difference between these two is very important, especially in chemistry, when you're dealing with sig figs, for example, significant figures, right? We want to round to a certain decimal place, or maybe we're looking at a bunch of numbers and which one's the most accurate, versus which one is the most precise, that sort of thing. So those instances are why accuracy versus precision is very important. Alrighty, next is experimental design. So experiments aim to test a hypothesis. A hypothesis is a proposed explanation of a phenomenon or just something that you test. Experiments have 3 variables: a control variable, which is what you compare your other variables to, your independent variable, which is the variable you manipulate, and your dependent variable, the variable that you measure. So for example, if we had an experiment where we wanted to test the effect of different genres of music on plant growth, our independent variable would be the different genres of music, our dependent variable would be plant growth, and our control variable could be plants that aren't listening to any music at all or aren't in an environment with music, because plants don't have ears, of course, but our control would be plants that are not in an environment with music. So this design can be explained over here on the left. It's kind of like a flow chart. So first we have to ask a question, all right? For example, do plants grow better when listening to music, right? Now we do some background research, and these are interchangeable, right? As you ask a question, you want to do research about it and see if there are any possible answers so far. But as you do research, you can come up with more questions as well and expand your knowledge and expand your questioning. After that process, you want to construct a hypothesis, right? So this is the proposed explanation. This can look like an if-then statement. Then we want to test using an experiment. This is when you actually carry out the process. Next, we ask, is our procedure working? If not, we can troubleshoot the procedure and carefully check all steps in setup, and then go back to testing with an experiment. We can restart, maybe alter the procedure a little bit, that sort of thing. But if it is working, we can go on to analyze our data and draw conclusions. All right. In this step, we can ask if the results align with our hypothesis. If they do, we communicate those results. If they do not, we still want to communicate those results. However, that experimental data can become background research for a new or future project, and you can ask new questions to form a new hypothesis and do your experiment again and start the process over. Ready. The next section we'll be going over is chemistry. The first topic within chemistry is atoms. Atoms are the smallest unit of matter. Atoms of a single type are called elements, such as carbon. Atoms of different types bonded together are called compounds. There are different parts of an atom. The nucleus, which is composed of the proton and the neutron, multiple of each usually. Protons have a positive charge, and neutrons are neutral charged particles. The electron cloud is where the electrons live. Electrons have a negative charge. So over here on the right-hand side, we have a picture. This is a carbon atom, right? In the nucleus here in the center of the rings, we have six protons and 6 neutrons. That is how we know that this is carbon, because there are 6 protons. The protons indicate which element it is. And as you can see, the protons have a positive charge. Neutrons, they don't have any charge in the middle. And electrons are surrounding it. Protons equal the number of electrons, except in ions. But this is not an ion, this is just an element currently. Next, we'll talk about the periodic table. So the periodic table is where all of the elements known to man are housed. Some of them are man-made, others of them are natural elements. Most of them are natural elements. They contain metals, nonmetals, metalloids, gases, what have you. The ones that have squares around them or rectangles here, they are very commonly used, and they are all gases. So each column is a different group, each row is a different period. Some of the groups, group 1 is called the alkali metals, group 2 is the alkali earth metals, group 7 is known as the halogens, and group 8 is known as the noble gases. Each group has a name, but these are just very common ones that elements are pulled from in various problems. But each column, each group or family, those elements react very similarly to one another. For example, group 8, the noble gases, are all very unreactive. They have a very low reactivity rate. Ready. So how to read the periodic table. So the atomic number is the number of protons. It identifies what the atom is. So over here on the right-hand side, we have carbon, has 6 protons, and that 6 identifies that this is carbon. The atomic mass is the protons plus the neutrons. Isotopes have the same number of protons but different number of neutrons. And the mass is the weighted average of all isotopes. So this 12.011 right here, that's the weighted mass average of all the isotopes of carbon. Elements share similar properties down a column. That is called a group or a family, as we talked about on the last slide. Stable elements have equal numbers of protons and electrons. Ions, atoms with charge due to unequal protons and electrons, can either have a positive charge or a negative charge. If it has a positive charge, that means it lost electrons and it's getting less negative. If it has a negative charge, that means it gained electrons and it has more negative charge. So this is just pointing to each one of the elements that we talked about within carbon. So this is the atomic number is 6, the atomic mass is 12.011. Next is bonding. So bonding is how elements combine using their electrons. Ionic bonding is when a complete donation of an electron from one to another occurs. Example, NaCl, table salt, right? Then covalent bonds is the sharing of electrons between two elements. These can either be nonpolar or polar. If it's nonpolar, that's the equal sharing of electrons, but if it's polar, that's the unequal sharing of electrons. So there are different types of reactions in chemistry. Chemicals react to form new compounds. So this chart shows us our kinds of reactions, what they are and what they mean, and then their general formula. So a combination reaction is when two or more compounds combine to form one compound. The general formula can look like A plus B yields, because the arrow means yields, AB. A decomposition reaction is the opposite of a combination reaction. A complex molecule breaks down to make simpler ones. So AB yields A plus B. It's just the combination reaction flipped. Precipitation reactions are when two solutions of soluble salts are mixed, resulting in an insoluble solid or precipitate forming. This formula can look like A plus the soluble salt B yields precipitate plus soluble salt C. The next type of reaction is a neutralization reaction. It's when an acid and a base react with each other. Generally, the product of this reaction is a salt and water. So this general formula can look like acid plus salt yields salt and water. A combustion reaction is when oxygen combines with a compound to form carbon dioxide and water. These reactions are exothermic, meaning they give off heat. So this formula can look like A plus oxygen, elemental oxygen, yields H2O, water, plus CO2, carbon dioxide. The last type of reaction we'll talk about here is displacement reactions. This is when one element trades place with another element in the compound. This general formula can look like A plus BC yields AC plus B. As you can see, B and C just switched places. Ready, next we'll talk about dilutions. This is when the concentration is constant, but the volume changes. Over here on the right graphic, we have a stock solution before dilution. Stock solution just means it's the original solution. So right here, we have a little bit of water, and the two beakers contain the same number of moles of the solute. So they contain the same amount of the solute, which is represented by the purple balls here. Now we add more water, and the solute is more spread out, but it contains the same number of molecules or the same number of moles of the solute, right? So we just add more water. This is called dilution. And if we need to find one of these variables, if we are given a concentration and a volume of the first solution, and we want to find the concentration of the second solution, but we're also given the second solution's volume, we can solve for that using this equation. The concentration of the first solution times the volume of the first solution equals the concentration of the second solution times the volume of the second solution. Let's say we were trying to find that concentration of the second solution, as I stated before. We would do the concentration of the first times the volume of the first, and in order to solve for this variable here, we would divide that by the volume of the second solution. Next is talking about pH. So the pH scale is shown over here on the right-hand side. It contains acids and bases and more neutral substances as well. An acid is any compound that releases H plus ions when in water. They release hydrogen ions. A base is any compound that releases OH negative ions when in water. pH measures the concentration of hydrogen ions in a solution. So over here on the right-hand side, we have our acids, which are all the way to the left, and bases, which are to the right. In the center, we have more neutral solutions, such as water, urine, seawater. An example of an acid is battery acid, whereas a base can be drain cleaner. The lower the number, the more acidic it is. The higher the number, the more basic the solution is. Ready. Next section we'll go over is biology. So macromolecules first. The major molecules that compose organic creatures are carbohydrates, also known as sugars, lipids, also known as fats, proteins, nucleic acid, such as DNA and RNA. So now we'll look at DNA versus RNA, some similarities and differences. Ready? DNA, known as deoxyribonucleic acid, is used for genetic information. It's in the form of a double helix. So if you look at this image right here, DNA is on the right-hand side. It has a double helix shape. It contains thiamine as a base, and it is stored in the nucleus of the cells. Whereas RNA, ribonucleic acid, is used for protein synthesis. It has a single string of nucleic acids, as seen in the image right here on the left. It's just a single string. It's not the double helix like DNA. It contains uracil as a base instead of thiamine. So over here, it contains cytosine, guanine, adenine, and uracil, whereas DNA contains cytosine, guanine, adenine, and thiamine. And it is only found in the cytoplasm of the cells. Next is cell structure. So the cell is the smallest unit of life for any organic creature. Organelles of the cell have specific functions. So over here on the right-hand side, we have an animal cell pictured. There are different organelles within this cell, such as the mitochondrion. We have ribosomes, lysosomes, centrioles, centrosomes, peroxisomes, Golgi apparatus. They each have a specific function that help to carry out the life of the cell. And here is the nucleus. So this is where the DNA would be stored. This whole thing right here is the nucleus. The smaller portion is the nucleolus. There's a nuclear pore, which are just holes so that it's semi-permeable. Then there's nucleoplasm around the nucleolus, and then the nuclear envelope is what encloses the whole nucleus. Ready, next is cell division. So the cell cycle has multiple steps. First is G1, which is the growth phase 1, synthesis phase, which is S phase, G2, known as growth phase 2, and then mitosis, which is known as cell division, and cytokinesis, which is cell splitting. It is a common misconception that mitosis is the entirety of the cell cycle, whereas it's only a portion of it. So the image over here lays this cell cycle out for us. So first we have G1, which is the growth phase. This is when cellular contents, excluding the chromosomes, are duplicated. If the cell is deemed unfit to undergo the cell cycle, it enters G0, which is cell cycle arrest. Next, the S phase, the synthesis phase. Each of the 46 chromosomes are duplicated by the cell. Next is G2, growth phase 2. The cell double checks the duplicated chromosomes for error, making any needed repairs. G1, S, and G2 are known as interphase. Then mitosis, which is cell division, and cytokinesis, which is cell splitting. So as you can see, mitosis is only a fraction of the cell cycle. As we see here, these are different steps in mitosis. Mitosis forms two identical diploid or daughter cells. Meiosis is mitosis happening twice, but forming 4 haploid or half cells. The first step in mitosis is interphase. Ready? This is known as G2. Then prophase. We start to see an early mitotic spindle and the 2 sister chromatids. Then prometaphase. The kinetochore microtubules are seen, and the fragments of the nuclear envelope, as well as the kinetochore. Then metaphase. Chromosomes lined up at the metaphase plate. Then anaphase. The daughter chromosomes are pulled apart to the ends of the cells. Then telophase and cytokinesis. We start to see a cleavage furrow, which is the little divot here between the 2 parts of the cell, and the nuclear envelope starts to reform, but twice because there's now going to be 2 cells. Next, we'll go over Punnett squares. So Punnett squares are used to predict the genotype of offspring, resulting in a specific phenotype. Genotype is the letters or the gene, and phenotype is the physical characteristic that it portrays. Allele pairs from your parents, one from mom and one from dad, are given to each child. Human genome consists of 46 chromosomes, 23 allele pairs. The possible outcomes are homozygous dominant, 2 capital letters, heterozygous, 1 capital, 1 lowercase, and homozygous recessive, 2 lowercase letters. So over here on the right-hand side, we have an example of a Punnett square. Let's say the top here represents dad, and the side represents mom. We got a capital T and a lowercase t from dad and a capital T and a lowercase t from mom. They are both heterozygous, which means that 1 capital, 1 lowercase. The dominant trait represents the capital letter, and the lowercase letter represents the recessive trait. Right. So how you set this up is you put one letter above each box, and on the side, one letter beside each box. Now you take the two letters, so the one on the top and the one on the side, and you put them together in the box. And the same thing for the next one. We always put the capital letter first. So the capital T and lowercase t. Same for this one, capital T, lowercase t from the top and the side. And then the top and the side, these are both lowercase t's. So as we can see, we have one homozygous dominant, two heterozygous, and one homozygous recessive. So 75% or 3/4 of the offspring could portray the dominant trait. Ready. Next is talking about respiration. Taking a fuel source inside the mitochondria, glucose, and converting it into ATP, adenosine triphosphate, the main energy molecule of the human body. This is known as cellular respiration. And I'm sure you all talked about it a lot in your biology classes. Ready? This is the equation for it down here. So we have glucose, which is C6H12O6. Added to oxygen, 6O2. Yields, arrow means yields, 6CO2, which is carbon dioxide, plus 6H2O, known as water, plus ATP energy, right? And this is the balanced equation for cellular respiration. Next is taxonomy. This is how animals and plants are categorized. An animal is called by its genus and species. So over here on the right, we get really, really specific as we go down. So the first is the domain, which isn't pictured here, but that's the first box, domain, then kingdom, phylum, class, order, family, genus, and species. So right here, this panther, the kingdom it belongs to is Animalia. Then the phylum is Chordata. The class is Mammalia. Order is Carnivora. Family is Felidae. Genus is Panthera. Species is Panthera pardus. So we would call this by its genus and species. So Panthera pardus. And if we need an acronym to remember this by, you can use do keep ponds clean or fish get sick. So we can remember that so that we know the order as we get more specific. Next is anatomy and physiology. The first topic covered here is with enzymes. Enzymes are catalysts who speed up chemical reactions by lowering their activation energy. Anything that ends in ACE, ACE, is an enzyme. Examples include lactase or amylase. They bond to specific substrates like a lock and key system. So over here on the right, we have a substrate entering active site of the enzyme. This is the substrate, and these are the active sites of the enzyme. Next, the substrate/enzyme complex. The enzyme changes shape slightly as substrate binds. Then our enzyme or products complex. And then the products leaving the active site of the enzyme is pictured in the last image here. The products leave the active site of the enzyme. Ready? Next, we'll talk about vitamins. So vitamins are necessary for various biological functions. There are 13 different vitamins, either being water-soluble or fat-soluble. So over here on the right, we probably all recognize this, right? Flintstone gummies. The vitamins provide different functions for our body, helping to either restore vitamin stores, or they sometimes help when you're sick to replenish your nutrients, but they are very important and you should know the different functions of different vitamins. Ready. Next, we'll talk about anatomical positioning. So here we can see that we have different planes. So these walls drawn through the person. This person is in anatomical position. Their eyes are forward, head is level with the ground, palms are facing forward, feet are apart, their back is straight. So now we can talk about the positions and the words that go along with where we're talking about on the body. Lateral means away from the center of the body. Medial means towards the middle of it. Ventral is towards the front. This is also known as anterior. Dorsal is towards the back. This is also known as posterior. Cranial means towards your head. Caudal means towards your feet. The transverse plane cuts your body in half horizontally. The sagittal plane cuts your body in half vertically. And the coronal plane cuts your body between ventral and dorsal, so from front to back. Righty. Next, we'll just talk about some regions of the body. So the cephalic portion is your head. Cephalic means head. This includes the frontal, so kind of like your forehead, orbital, around your eyes, nasal, buccal, and oral. Next is the cervical region, near your neck. Thoracic region, which contains the sternal and axillary regions. The abdominal region, which contains the umbilical region around your belly button. Pelvic regions, inguinal, which is also known as the groin. Pubic, also known as the genital region. Then we have our upper limbs. So from top to bottom, we have acromial, deltoid, brachial, antecubital, olecranal, antebrachial, which also known as your forearm, and carpal, then digital, which is your fingers. The lower limbs, coxal, also known as your hip, femoral, where your femurs are located, patellar, it's in the kneecap region, popliteal, back of your knee, crural, kind of like the top of your shin, sural, more like your calf, fibular, where your fibula is located, tarsal, known as your ankle, calcaneal, your heel, digital, your toes, and plantar, which is the bottom of your foot, right? So the occipital is the back bottom portion of your head. Cervical is the back, also the back of your neck. Then we have the back. So scapulars near your scapulas. Vertebral, which corresponds to your spine. Lumbar, sacral, and then gluteal, which is your behind. Ready. So anatomical systems, skeletal system, muscle system, nervous system, endocrine system, cardiovascular system, respiratory system, digestive system, and immune system. For each of these systems, you must know their general function. So what does it do and how does it do it? So how does the skeletal system carry out its function? How does it provide us support? Right? Next, the diseases of the system. So what happens when the system malfunctions? So for the skeletal system, there's osteoporosis, right? What does that mean? And what happens to the body during that disease? Then the major structures of the system. What's necessary for the system to function? So this, for the skeletal system, can look like the different bones of the body. How are the different fossas and fissures? How are they related to keeping the body functioning? How are they related to holding the body up, protecting it? Ready? Now, if you have any questions, you can ask them below, but I hope you all do very well on your exam. Make sure to study the topics in here and more in depth. But good luck on your exam, and I hope you have a wonderful day.